Yeah, my name is Patrik Flammer. I'm an editor for Genome Medicine and BMC Medical Genomics, and I'm joined here by David Cortez from Vanderbilt University in Nashville. Thanks for taking time to have a chat about your research, and yeah, maybe we start with how you got into the field of like DNA repair and.
Sure. Well, it's a pleasure to be here. About 20 years ago is when I got into this field, a little bit more. I had done my PhD at Duke studying tyrosine kinases, and saw a seminar by Steve Elledge. And he had lunch with us as students, and I thought his research was really interesting. So I applied to him for a postdoc and moved to Houston, where he was at the time, at the Baylor College of Medicine, and started working on DNA damage responses, the ATR kinase, and how it sensed problems and sort of substrates of the DNA damage response. So and then in 2000, that was a little over 20 years ago, and then 2002 I started my own laboratory at Vanderbilt University.
All right.
Yeah.
So you're specializing more on the replication fork.
Yeah. So initially, I started my lab on the ATR kinase, how it was regulated and how it was activated, and we continue to do a little bit of work on that. And ATR is a primary sensor of replication stress. So it's activated every S phase whenever there's a problem at a replication fork. Yeah. And it does a lot of different things to signal for DNA repair, damage tolerance. Sure. And so lately, actually, while we continue to work on ATR, my lab really changed directions a little over 12 years ago or 10 years ago, when we started doing proteomic screens looking at proteins that were at replication forks. Yeah. And a student of mine invented a technology she called iPond, and that led us to purify replication forks under all kinds of stress conditions or genetic changes, and identify lots of cool proteins. And pretty much since then, we've been working on those proteins that have come out of that proteomic screen.
Yeah, so sometimes funny how like one project that was a sideline sort of takes over, and
yes.
When you see a lot of interesting things coming out of that.
Yeah, I'd like to pretend I was really smart and thought of this idea and thought, oh, this would be great to do exactly what we did, but it came out of actually a small question we wanted to answer, and we were trying to develop a method to answer a small question, and then realized after we developed a method that, hey, this could be useful for a lot of things. And that's really just changed the direction of my laboratory. And I'm really actually most pleased that many people in the field have used it, and it's been beneficial to lots of people to study replication and replication stress related things, and even chromatin deposition and things like that during the replication process.
The smart thing is that you actually realize that it has more users than just the one small problem, right?
Yeah. Yes, it was good that we figured that out. Yes. I have to say that the challenge with it actually is to know when to stop doing the proteomics and to get on with the mechanistic biology and mechanistic science of it. I think we've done a fairly good job of balancing those two things. Certainly there's more things to learn and identify, and the proteome at the replication fork is certainly not a complete list that we know everything. But we have a very good idea now of, I think, all the players that can be involved in doing DNA replication and damage tolerance and replication-coupled repair.
Yeah, okay. So the technique you mentioned, what exactly was the idea behind it?
It's rather simple. So at the time, we were trying to figure out a way of actually purifying the MCM complex and associated proteins, because we had a specific question about when they were being phosphorylated. We knew the ATR could phosphorylate MCM2. Yeah. We wanted to know, is that happening at elongating forks or at origins of replication? Yeah. The two things are very different, because if it was phosphorylated at origins, that would imply it was regulating initiation. If it was phosphorylated at elongating forks, then it was regulating the helicase activity itself and elongation. So the IPON technique really was our method to purify elongating replication forks. Instead of purifying the proteins directly, the method purifies the nascent DNA.
Oh, right, okay.
Yeah, so we label cells. At the time, EDU had just started to become available as a reagent. We labeled cells with EDU for short periods of time.
Yeah.
And then that provides us, we can put a handle on the EDU using click chemistry, put a biotin group on it. And that then allows a very simple single-step purification of the nascent DNA.
Yeah.
So if one does that and does it under different conditions, you can actually differentiate the proteins really bound to the nascent DNA and sort of adjacent versus those that are just in bulk chromatin everywhere.
Yeah.
And we really combine it now with really quantitative mass spectrometry approaches, SILAC-based mass spec mostly. And it gives us a good handle on how does the replosome change, what proteins come in when there's a problem. If we delete a protein, what happens, delete a gene and say, well, how does that alter the replication fork proteome? And it's really a good starting point for projects.
Yeah, I guess it allows you to really interrogate all the components, like what happens if I take that one away, or if that one doesn't work, or if there's any like other problems with the replication.
Yep.
Yeah,
that's exactly right. That's the right summary.
Yeah, it sounds like a very versatile method, really.
Yeah. So, and then what it led to was all these proteins that were basically unstudied. So one I'll talk about at this meeting is a protein we named RADX. And it was annotated at the time as CX or 57. So an open reading frame on the X chromosome. Nothing known about it, but we knew from our IPON experiment that it was at replication forks. And furthermore, it was becoming more abundant in response to replication stress. And there's a variety of proteins like this that we saw in our data set, and we said, well, what is that? Why is it there? When is it there? And then we do all of the sort of detective work after that to figure that out. What does RADX do to promote accurate replication? Which is really the basic question of my laboratory, is how does a cell complete DNA synthesis, copy 6 billion base pairs of DNA, do that tens of trillions of times during our lifetime without creating so many errors that, they do happen, errors happen, and that can cause disease, developmental disorders, aging, cancer. So largely we're trying to figure out how does that happen properly, and sometimes what we can do about it if it doesn't happen properly. And RADX is one of those proteins that is involved in that process.
So you're also looking into sort of drug development, or is that
indirectly.
Too far? Indirectly.
Yeah. So my motivation has always been basic discovery. I've always just really wanted to know how things work. That being said, I'm part of the Vanderbilt Ingram Cancer Center. I liaison with lots of clinician scientists who are very interested in using the knowledge that we create in the laboratory to prevent or treat cancer. And it is very valuable knowledge for that. And I think, none of the advances we've made in cancer treatment would have been possible without these basic science discoveries. Sometimes we don't know which ones are important. And we still actually don't know how important RADX is for disease. I don't know if it's really contributing to disease. I don't know if it's a good drug target. Might be, might not be. But without understanding it in detail, we'd never figure that out.
But that it's involved. So you mentioned that it changes in concentration.
It gets more. It's recruited to stalled forks because it's an interesting protein. It actually likely evolved from RPA, which is the major single-stranded DNA binding protein, and so it looks a lot like the largest subunit of RPA in domain structure. RPA is a heterotrimer. RADX is more of probably a homodimer, is what we think now. But like RPA, it binds single-stranded DNA, and so that's probably why we observe it whenever we stress cells with an agent that damages DNA or impedes replication. Single-stranded DNA becomes more abundant. RADX binds that single-stranded DNA, and that's probably why we identified it through our IPON method. And then our task for the last 5 years since we identified RADx has been to figure out what it does. Why is it there? Same with all the other proteins that we've discovered. It's a crazy protein called HEMCs that we discovered, and ETA1 is a ATR regulator we discovered this way. But all of these, once we find out they're there and how they get there, well, what do they do?
Yeah.
Why are they important to promote DNA synthesis in an accurate way?
Yeah, because they must have an important function if they get recruited.
Yeah, so currently our model for RADx specifically, and I'll mention this tomorrow, is that it regulates this process called fork reversal, which is a really interesting gymnastics that the replication, that happens at replication sites as a DNA damage tolerance mechanism. But it's a very dangerous process as well because it creates a double-stranded DNA end that can be processed. It creates a sort of something that looks like a holiday junction that can be processed, and these things can create breaks if they're processed improperly, or degrade the DNA if processed improperly. So the fork reversal process itself is highly regulated. And what we've discovered is RADx is one mechanism of that regulation. It really confines fork reversal to only happening when the fork is persistently stalled, and doesn't allow it to happen when the fork is naturally elongating.
Okay, so that's the fork reversal only happens when there's problems with the replication.
That's the expectation. That's the hope.
Right. Okay.
So in fact, what we find is if we delete RADx, then cells accumulate reversed forks even in unstressed conditions, and those tend to break. And so we get increased double-strand breaks in RADx deficient cells. All of that's traceable to excessive fork reversal because we can actually rescue RADx deleted cells by inactivating fork reversal. So RADx is in that situation is preventing reversal from happening.
So that might be a quite important function of the RADx protein.
Yeah, I think that's likely a major function for it. It may have functions outside of that as well. Just like RPA is a single-strand DNA binding protein that has functions anytime there's single-strand DNA, RADx may have multiple functions. One thing that's interesting about it is it also binds this repair protein, RAD51. And RAD51 is arguably the most important repair protein in our cells. It's the recombinase that does homologous recombination. It functions to do double-strand break repair. It also is involved in fork reversal. So all of our data so far has been tracing RADx function in the context of its regulation of RAD51. And largely at forks. But since RAD51 has other functions, it wouldn't surprise me that RADx also is regulating it in those other situations. We don't have any direct evidence of that. That's pure speculation at this point. You know, we study it where we know best. And then sort of the context that we
branch
study
out,
it in. Absolutely. Or other people can.
You
know, that's the beauty of science.
It's about the sharing.
Exactly right. That's why we come to these meetings, right? I learn a lot from my colleagues. Hopefully something I say will spur my colleagues to do the next great experiment and learn something new that we couldn't have done ourselves.
It's sometimes just about sharing ideas or triggering ideas, and a bit like with in your personal story when you said that you heard a seminar and then sort of started getting interested in a different field. That's
right. That's exactly right. And this is one thing I really like about science is I like that it builds. It's sort of a large team throughout the world, right? There's competition, right? That I think that when it's healthy, it's fine, right? I think that's okay. But largely, it's the cooperation that I really appreciate. Cooperation through ideas, sharing of data, sharing of suggestions. You know, some of the actually, you know, some of the best discoveries I've made, both as a trainee and then in my own lab, have come because somebody suggested something to me. They suggested an idea. And some of their ideas, of course, aren't right. But every once in a while, somebody comes from a different perspective, and they see the data I present, and they'll give me a new idea that then pans out into something really important. And so that sharing of information is critical. I really do enjoy that.
Yeah, I agree. It's sometimes you need to take a step back from what you do, and just to see the greater picture, or have
Well,
also have a crazy idea sometimes to just try things. And
also different expertise.
yeah,
So
absolutely.
I mentioned briefly one of the proteins we identified in IPON is a protein called hemces. It doesn't really matter what it does, but we were struggling. We thought it was interesting, but we had done all of the kinds of experiments we knew how to do. So I presented our studies to an internal group at Vanderbilt and a chemist, a nucleic acid chemist, but a chemist. He's not a biologist. He doesn't know anything about replication, but he knows a lot about DNA. And he was there, and I was presenting this, and he's a little bit older than I am. I was presenting this data, and he said to me, well, have you looked at this protein in the context of abasic sites? Now, abasic sites are one of the most common lesions in our cells. Thousands of them happen every day. I said, abasic sites, that's boring, right? We know how abasic sites are repaired.
Yeah.
Basic scission repair deals with them very easily. But he suggested it, and sure enough, this protein turns out to be a new processor, a new regulator of abasic site processing and repair. And the key there was the sharing of information before it was published, but also that he had totally different expertise than I did.
Yeah.
And the key for him was that he knew literature from the 1980s that I didn't know, because I have to admit, I haven't read a lot of 1980s literature, 1990s literature. Yeah. Most of my, formative experiences in science came, in the late 90s and afterwards. And so just that different expertise, it was essential. Yeah. And at this meeting, that's what happens, right? Yeah. We talk to our colleagues. They have a different approach, a different set of experiences, and then we learn something new.
Yeah, that's a great thing about meeting people. Actually, and it's great that we actually get to do that in person again.
Oh yes,
because it's
it
not
means
the
so
same.
much more. Yeah. I learned so much from having breakfast with somebody or just bumping into them at coffee break and talking about something. Those types of experiences do not happen over Zoom. No. You can't really remake those things.
It's not spontaneous.
Yeah.
So even if you've got a breakout room, and I as an editor, it's even worse because here I can mingle and we can have a conversation, and you can spare 5 minutes over coffee, but over Zoom it's not quite as easy. It's not quite as easy just to meet people and to talk about research.
Yep, I completely agree. Yeah, and because I've been in the field for a couple decades now, I have a lot of friends that I like to see and catch up with too.
Yeah, that's
and those personal interactions are really valuable, and doing those over Zoom it's not the same.
It's not, no. It can, we can make do if we have to, but
right.
Yeah.
Yep. Yeah.
Right.
We're almost near the end.
Yeah.
But, I can preview for you actually the unpublished stuff I'm going to tell everybody tomorrow, if you'd like to hear that.
Yeah. Oh, yeah. That would be.
So I mentioned RADX and fork reversal. Yeah. What that actually led us to is, since we know RADX regulates RAD51.
Yeah.
We wanted to know, well, how does RAD51 work? We can't understand a regulator without understanding RAD51. And we have a model, which I admit is preliminary, but that's why we come to meetings to present it. But the model is that RAD51 at forks is doing very similar things that it does at breaks. Yeah. And that is, it's doing strand exchange. And at forks, what we think that the reason it's doing that is the helicase complex is still present on DNA when a fork stalls. And fork reversal, the way everybody draws it, it's like migrating this junction backwards. But how does that happen if the helicase is present? I mean, it's occluding the place where these fork reversal enzymes are supposed to bind and work. And so our model is that RAD51 creates a new junction. Not the same fork junction, but a new junction behind in order to allow these fork reversal enzymes to bind and do branch migration and actually allow fork reversal to happen. So this is a model where we've generated a little bit of data that I'll present that is consistent with it, but it answers some of the major questions in the fork reversal field as well. What does RAD51 do? What happens to the CMG helicase during fork reversal? How can this possibly happen if you've got the helicase present? Does it have to unload? No, it doesn't unload, because if it unloaded, there's no way of getting it back on DNA during S phase, at least that we know. And so our model really solves a lot of these problems and answers these questions. And we've got some really, I think, intriguing data that supports that model that I'll share with everybody, and we'll see how people react.
Yeah, it sounds very intriguing, and it's definitely the sort of approach that you've got. You need to first figure out what one protein does if you can't figure out you can't figure out what all of them do at once, right?
No, you can't. And you certainly can't figure out how something's regulated until you know how the actual machinery works by itself. Figuring out the regulation process is complicated, but really almost impossible without knowing how the machinery of this process works itself.
The regulation is not necessarily straightforward, or can be different under different conditions.
Oh, this is the fascinating thing about RADx is I told you earlier that RADx inhibits RAD51 in fork reversal, but that's only true in unstressed cells. When we persistently stall forks, RADx turns around and actually promotes fork reversal. It switches function. It looks like it switches functions, and it still does that through binding RAD51. And we have a really interesting model of how it does it. Actually, I think it does exactly the same thing biochemically at all times. But you said, well, the context matters. And that's what we think is different. Is the context of an elongating fork is very different than a stalled, persistently stalled fork. And so we have this crazy model of how RADx can possibly switch functions without actually changing its biochemical activity at all, through essentially making RAD51 filaments dynamic and unstable. And that, we think, is what's important for fork reversal. But by doing that in the context of an elongating fork, RAD51 never gets enough hold onto it to do the reversal step. But at a persistently stalled fork, it's not holding on too tight. It's balanced because RADx helps it balance its function, and that allows it just the right amount of activity to promote reversal. Admittedly, this is a little bit speculative, but that's what we think is happening.
But there's no change in conformation or
anything. So we can't find any change in the binding partners for RADx or post-translational modifications. We don't have a really high resolution structure yet. We're working with collaborators to do that. That would be great. So there's other possibilities. But my favorite is the simplest. I always like the simplest idea. My simplest idea is RADx does what RADx does. It's always doing the same thing. And it appears to switch functions biologically because the context in which it's functioning has changed.
Yeah, it's definitely, it makes much more sense. Sort of like it's more like an Occam's razor.
Well, that's part of it. No, that's why it's appealing to me. But as you know, biology is complex. Sometimes it doesn't make any sense. The way things evolved, they don't make sense. So we have to allow for that possibility as well. But that's what I'll talk a little bit about tomorrow. And as I say, my colleagues will come and they'll ask questions, and hopefully
trigger some new ideas.
New experimentation, and we'll either figure out that we're all wrong and move on to the next idea, or we'll continue and help prove this model or at least support this model.
Yeah, you can't really move forward until you tried some of the wrong ideas first either.
Yeah. Well, it's a pleasure talking with you.
Yeah, it's great talking to you. Thank you for taking the time and for your insight into your research.
Yeah, great.
Great.
Thanks.
Thank you.